So in today's video we are going to study about cell membrane, and this cell membrane we know it is also known as cytoplasmic membrane or plasma membrane. Plasma membrane because it covers the protoplasm of the cell. So let's get started. We will first discuss about the discovery of cell membrane. Okay, how it was discovered. It was discovered by Schwann in 1838, and this Schwann was working on zoology. He was a zoologist and he was working on animal tissue. He was the first person to identify that animal cell do not contain cell wall but only a cell membrane. Cell membrane, that is, the term cell membrane was first used by Nageli and Kramer in 1855. Now this cell membrane term was used by Nageli and Kramer in 1855. In 1931, that is exact 80 to 90 years later, it was named as Plasmalemma. Okay, the same plasma membrane is named as Plasmalemma by Plove. So he was the person to use term Plasmalemma. So what is plasma membrane? It is a thin membranous structure which covers the protoplasm of the cell. It is a membranous covering of protoplast. So this membrane will occur outside the cell. Okay, it will cover the cell. It is a covering of cell, and it is also the covering of cytoplasmic organelle, all the cell organelle. They are also having some kind of membrane, and this membrane are same as plasma membrane, same as biomembrane. Okay, so the word plasma membrane, biomembrane, cell membrane, cytoplasmic membrane, plasmalemma, all are synonymous and they are all same thing. Okay, now the biomembrane was found to be tripartite or trilaminar. What is tripartite or trilaminar? It is divided into three sections. Okay, now we will study the structure. There only we are going to discuss about this term tripartite or trilaminar. Let's see what is cell membrane composed of. Composition of plasma membrane. Now plasma membrane consists of lipid, protein, carbohydrate and water, and I have also written the proportion. See, the proportion is given from 20 to 79% lipid. Now lipid proportion in the cell membrane depend upon the type of cell it is. Okay, if it is RBC or it is another kind of cell, it is a muscle cell, or it depends on what kind of cell it is, and on the basis of that the proportion of lipid protein will vary. Okay, so now let us see. Lipid is around 20 to 79%. What is the lipid? What kind of lipid does it contain? It contains phospholipid, it contains glycolipid, then sterol, sphingolipid. Okay, so this kind of lipid it will contain. Then the nature of lipid is amphipathic. Lipids are amphipathic in nature. Okay, the lipid present in cell membrane or plasma membrane is amphipathic. It is having both a polar head and non-polar tail. It is something like this: polar head which is phosphate containing, polar head and non-polar, or you can say hydrophobic tail. This is fatty acid chain tail. Okay, it is containing carboxyl group. You know what is fatty acid, na? It is a hydrocarbon chain containing carboxyl group at the end. So this is a molecule, lipid molecule. Here it is having polar head which is hydrophilic in nature. It is polar in nature, and the tail is hydrophobic. it is fatty acid tail. So if it is fatty acid, then it is non-polar, or you can say it is a hydrophobic. Okay, it will go away from the water, it will move away from the water. So it is containing amphipathic lipid molecules. Then protein. Now proteins are also classified on the basis of its structure and its function. On the basis of structure, you can see proteins are of two types. Integral protein, those protein which are present in the membrane, it is spanning the membrane. Okay, it is present between the membrane and it's crossing the membrane. It is integral protein and it is a transmembrane protein, integral protein or transmembrane protein. And those protein which are present at the periphery, okay, it is not inside the membrane, so it is present on any side of either sides of the membrane, then it is a peripheral protein. So on the basis of position of protein, it is classified integral and peripheral. On the basis of its function, protein is classified. If it is enzyme, some kind of enzyme, you know biomembrane contains lots of enzyme. If you will see mitochondria, then its biomembrane will contain enzymatic respiratory enzymes. All the respiratory enzymes are present. They are proteinaceous in nature. We all know that enzymes are protein in nature. And transport proteins are there. So many protein. Yes, the main function of plasma membrane is transportation, transport of a small molecule, okay, through channels or through transport protein. So transport protein is also there. There are many protein which are forming the structure, so they are structural protein. So on the basis of function, the protein is also classified: structural protein, transport protein and enzymes. Okay, carbohydrate, generally oligosaccharide. Oligosaccharide, that is, what is oligosaccharide? It is 10 to 15 sugar polymer is known as oligosaccharide. So 10 to 15 sugar together will come and they will attach to some space, and that is known as carbohydrate or oligosaccharide. Either they will directly attach to lipid portion on the polar head of the lipid, and they will be known as glycolipid. Their name will be glycolipid if they are directly attached to the polar head. But if they are attached to some protein, some peripheral protein head, then they will be given name glycoprotein. So carbohydrate is present in form of hexose, that is your fructose, mannose, galactose, such kind of sugar will be present. Then water, around 20% of water it will contain. Okay, so plasma membrane contain lipid, protein, carbohydrate and water. Now many from year 1925, the many different researchers worked on the structure of biomembrane, how the biomembrane looks like. Okay, and they gave many models. Okay, two models were very famous: sandwich model and mosaic model. But the most accepted, widely accepted model is the mosaic model, which is a recent one. Okay, it came in 1972. It was given by Singer and Nicholson. Okay, Singer and Nicholson fluid mosaic model was the best model, and it was accepted. It was most widely accepted model about biomembrane. It shows that how the structure of biomembranes looks like. Okay, and before discussing about fluid mosaic model, I would also like to discuss about Daniel-Davidson and Robertson model. These models come under the sandwich model. So now the lamellar model or sandwich model, the Daniel and Davidson model, it was known as Daniel-Davidson model. James Daniel and H. Davidson were the first to give this model. It is also known as popularly sandwich model. Why sandwich? Like how sandwich is made, the two layers and in between there is a filling. So in this way, Davidson and Daniel, they showed that lipid by layer, there are two layer of lipid, and it is covered with protein, and that protein is also in globular structure. Okay, that is globular structure protein has covered both the side and both the side of phospholipid bilayer. Okay, so this was known as a sandwich model. Now Robertson model, he gave the model named unit membrane. He said that it is unit membrane. He modified the same Davison model and he said that it is phospholipid bilayer, and phospholipid bilayer is having a protein on both the either side. You can see the extracellular side is having a glycoprotein like this, and this cytoplasmic side is having some protein. So he also gave the same he also modified that model and said that it is unit membrane model. And by unit membrane, he worked on a plasma membrane of RBC under the electron microscope, and he gave the concept of unit membrane, which means that all the cytoplasmic membrane is having the similar structure of the 3 layers. Okay, and the lipid bilayer is sandwiched between the 2 layers of protein, and all the biomembranes, whether it is cytoplasmic membrane or it is organelle membrane, they all are made up of this unit membrane. Either they are present in single unit membrane or they are repeated in it depends. If it is a cytoplasmic membrane, then it is a multiple of unit membrane. So he said that this membrane is tripartite or trilaminar membrane. Okay, tripartite, that is lipid bilayer is your single, and one is protein, and this one is also protein. So he said that this is a tripartite, that's a trilaminar membrane structure. Now after this, the most famous, or you can say widely accepted, fluid mosaic model was given by Singer and Nicholson in 1972, and this model is also known as recent because it was given now only in 1972, some 50 years back. So according to this model, the membrane do not have uniform deposition of protein and lipid. So there is no uniform deposition. Okay, protein and lipid are present in form of mosaic anywhere. Okay, you know what is mosaic distribution. Okay, mosaic distribution means they can either present here or here. So it depends. It is not that they are going to be uniformly spread somewhere. Okay, and this word mosaic is very accurate as compared to other model. And then Singer Nicholson also suggested that in this model the membrane is quasi fluid in structure. Okay, it is not a solid, it has some kind of movement also within. Okay, they are having a flip-flop movement. They are this your lipid bilayer. Lipid can move upper side to lower, and lower surface, from lower surface to upper surface also. So this is a flip-flop movement. Okay, and there was lateral movement also. Here they can move either this way or this way. So it was moving also. So it was quasi fluid in nature. It was mosaic. Therefore it was named as fluid mosaic model. Okay, and they also suggested, not he, they, because 2 people were involved, Singer and Nicholson both were involved in this. So they also suggested that proteins are in form of integral or peripheral. So there are tunnel protein or transmembrane protein which are present within the membrane, spanning the membrane. So this was transmembrane protein. Okay, such kind of proteins are present, and some proteins are peripheral, they are extrinsic, they are present on the lipid bilayer surface, so they are extrinsic protein or peripheral protein. So they gave this about protein. They told us about protein, how they are present. And glycolipid, sugars are present either on the surface of phospholipid layer. If it is present on surface of phospholipid layer, then it was known as glycolipid, and if it is present on protein surface, it is known as glycoprotein. So glycoprotein and glycolipid together will form what? Glycoprotein plus glycolipid will together form glycocalyx. It will together known as glycocalyx, the most important term, glycocalyx. So, and there was one more thing, in fluid mosaic model there was asymmetry. So asymmetry, that is two surface of biomembrane are not similar. Okay, the membranes are asymmetric in nature. Lipid present on outer and inner side of bilayer are commonly different. Okay, there are some different type of lipid is present on upper surface, on lower surface there is some other kind of lipid is present, so they are asymmetrical in nature. And due to this this provide them multiple function. Okay, multiple function of biomembrane is due to this asymmetrical nature of lipid also. Let us see the function of cell membrane. Function of cell membrane, first of all, compartmentalization. Now this cell membrane is helpful in forming the compartment. It's going to separate one organelle or one cell from the other, so it helps in forming the compartment. Okay, so very first you can say function of cell membrane is compartmentalization, that cell membrane causes the formation of compartment, and this membrane, plasma membrane, either plasma membrane or a cell organelle membrane, they are going to separate that particular cell from the external environment, or an organelle covering can separate them from the other organelles, and it maintain their identity, individuality, or you can say internal environment, and help them to maintain their functional values also. Okay, second, protection. Obviously covering, it is a boundary, so it's going to protect it from other injuries, attack like this, any kind of stress. It's going to provide a protection from osmotic pressure also. It can tolerate some kind of osmotic pressure also, so it is kind of a protective layer. It protects from injury. Third, permeability. Yes, there are this membrane can be semi permeable also, it can be impermeable also. Impermeable means it will not allow any molecule or any solute or anything to move through it. Okay, so it is impermeable. If it is allowing some molecule or some fluid or some kind of molecule to move through it, it is semi permeable, it will work like a sieve. So there is a permeability also. Permeability, they are having a kind of pole like a section through which the solute or molecule can move. Then next is endocytosis. What is endocytosis? Endocytosis, as we all know, endocytosis and exocytosis, the movement of molecules to toward the interior of the cell is endocytosis, and the plasma membrane is going to work like a vesicle or something like that. It's going to form a vesicle and it will allow some molecule to get entered inside the cell. So that is the process of endocytosis, and here the plasma membrane also play the important role. It will contain many kind of receptor for the various hormone. We all know hormones cannot directly bind to somewhere on plasma membrane. There is some kind of receptor protein present on the plasma membrane. See, if it is a membrane, then there is a receptor protein over here, and the molecule, signal molecule, your hormone will come and bind to this receptor. So they possess receptor also. In the same way, they also contain transport channel, transport protein for the molecule entry. So Here it was a function of cell membrane. In this way the plasma membrane or cell membrane is there in the cell. I hope you understood about this, whatever we have discussed today about plasma membrane, and these all are important for the exams, for the different exam, either it is NEET or any other biological exam you are giving, and even in BSc notes also it is very helpful. So thank you.